What is the structure of enzyme? Tertiary structure, okay? So to understand this, we should study enzymes a little more. We have already understood the idea of an active site. We know that when the enzyme is in its tertiary state, so it has, since it is folded upon itself, so it has certain crevices or pockets, and we call that as the active site because those are the sites where the substrates would come and bind to. Now, the chemical or metabolic conversion refers to a reaction. The chemical which is converted into a product is the substrate. Now, hence, enzymes, that is proteins with 3D structures, have an active site, convert a substance or a substrate into a product P. Symbolically, it can be depicted as S is getting converted into P. And what is P? P is the product. Now, it is now understood that the substrate S has to bind the enzyme at its active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation of an ES, that is enzyme substrate complex. E stands for the enzyme. This complex formation is a transient phenomenon. Transient means something that occurs for a little period of time and then it ends. So transient or ephemeral is the same thing. Now, during the state where substrate is bound to the enzyme active site, a new structure of the substrate, which is known as a transition state structure, is formed. Transition state structure is exactly before the substrate gets turned into the product. That is the transient state structure. Very soon after the expected bond breaking or making is completed, the product is released from the active site. In other words, the structure of the substrate gets transformed into the structure of the product. The pathway of this transformation must go through the so-called transition state structure. Transition state, again, the period from which the substrate gets turned into the product. Now, there could be many more altered structural states between the stable state and the product. Implicit in this statement is the fact that all other intermediate structural states are unstable. Stability is something related to the energy status of the molecule or the structure. Hence, when we look at this pictorially through a graph, it looks something like this. It is a hyperbola. Now, the x-axis, the y-axis represents the potential energy, and the x-axis gives us the progress of reaction. Now we know that now they are showing two kinds of reactions, one with the enzyme, one without the enzyme. So if you see it for the one without the enzyme, the transition state is quite high, whereas the one with the enzyme, the transition state is quite low, or the activation energy that is required, it is low with the enzyme. So with this, we can understand that what is the enzyme doing? It is actually lowering the activation energy that is needed to proceed the reaction in forward direction. Now, the energy level difference between S and P. If P is at lower level than S, the reaction is an exothermic reaction. One need not supply energy by heating in order to form the product. However, whether it is an exothermic or spontaneous reaction on an endothermic or energy requiring reaction, the S has to go through a much higher energy state or transition state. The difference in average energy content of S from that of this transition state is known as the activation energy. Enzymes bring down this energy barrier, making the transition of S to P more easy. When the enzymes will not be present, the activation energy that is needed to proceed the reaction will be much higher than what is required. So that is why enzymes are so important. Now, talking about the nature of the enzyme action, each enzyme has a substrate S binding site in its molecule so that a highly reactive enzyme substrate complex is produced. Also, enzymes are very specific. For example, we have studied so many different enzymes for glycolysis, so many different enzymes for this thing, aerobic reaction. So we are seeing that each of those reactions that are occurring, they are occurring because of a certain name of enzyme, and that is how it happens. Now, the enzyme substrate complex is short-lived. and dissociates into its products, that is P, and the unchanged enzyme with an intermediate formation of the enzyme product complex. Now see, first the enzyme along with the substrate forms the enzyme substrate complex, then it forms the enzyme product complex, and then the product gets released and the enzyme is unused. This is the greatest thing, that the enzyme does not get consumed during the process. Now, the catalytic cycle of an enzyme action can be described in the following steps. First, the substrate binds to the active site of the enzyme, fitting into the active site. So first, the binding takes place. Now, the binding of the substrate introduces the enzyme to alter its shape, fitting more tightly around the substrate. Okay, meaning the enzyme now forms according to the substrate's shape. Now, the active site of the enzyme, now in close proximity of the substrate, break the chemical bonds of the substrate and the new enzyme product complex is formed. Later on, the enzyme releases the products of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. So, the enzyme ultimately is unused and it can be used a number of times. Any doubts up until now, students?
No, ma'am.
Okay. Now, let us talk about the factors affecting the enzyme activity. The activity of an enzyme can be affected by a change in the conditions which can alter the tertiary structure of the protein. These include temperature, pH, change in the substrate concentration or binding of specific chemicals that regulate its activity. So there are certain factors that actually affect the enzyme activity. Now the first is temperature and pH. Enzymes generally function in a narrow range of temperature and pH. Each enzyme shows its highest activity at a particular temperature and pH. That is known as the optimum temperature and optimum pH, meaning the enzyme will work at this particular pH, let's just say 15 degrees Celsius. Then it will work at that only. If the temperature gets higher, it can get denatured. If the temperature gets lowered, it can get, you can say, inactive. Now, activity declines both below and above the optimum value. Low temperature preserves the enzyme into a temporarily inactive state, whereas the high temperature destroys the enzymatic activity because proteins are denatured by heat. So you must remember this one thing, that temperature above than the optimum temperature, if that is provided, the enzyme will get denatured. If lower temperature will be provided, then the enzyme will get inactive. Now talking about the concentration of substrate, like how much substrate is there for the reaction. So with the increase in the substrate concentration, the velocity of the enzymatic reaction rises at first, meaning as the substrate is increased, the enzymatic reaction, the rate of reaction also increases at first. Now, the reaction ultimately reaches a maximum velocity, that is Vmax, which is not exceeded by any further rise in the concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzyme molecules to bind with additional substrate molecules. So it doesn't mean that if the enzyme has been provided, so more and more, if we like keep on adding the substrate, the reaction will keep on getting happening. No, it will only happen up till a certain point wherein the velocity has reached its maximum value, that is Vmax. The activity of an enzyme is also sensitive to presence of specific chemicals that bind to the enzyme. When the binding of the chemical shuts off the enzyme activity, the process is known as inhibition and the chemical is known as the inhibitor. There is a chemical, when it will bind to the enzyme, it will look like a substrate, but it will not be a substrate, and when it will bind to the enzyme, the reaction will stop or get inhibited. So this process is inhibition and the chemical that is causing it is an inhibitor. Now when the inhibitor resembles the substrate closely in its molecular structure and inhibits the activity of the enzyme, it is known as competitive inhibitor. For example, the substrate looks like this and so does the inhibitor. So if the substrate did not go and bind to the active site, this inhibitor will go and the enzyme activity will be stopped. So that is competitive inhibition or competitive inhibitor is this. Now, due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind, and as a result, the enzyme action declines. Now, the example wherein this can be utilized is, or it is seen, is the inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in the structure. So, succinate and malonate, they have a similar structure. But when the succinate does not bind with the dehydrogenase and instead it binds with the malonate, then that is a competitive inhibition. And where do we apply this? So these competitive inhibitors are used often in the control of bacterial pathogens. Okay, meaning bacteria require certain substrate to synthesize the protein. Instead of that, we provide a competitive or inhibitor. So because of that, the bacteria would die. So this is how the enzyme activity is there. Any doubts up until now? Also, one more thing. The point wherein the maximum velocity has reached half its value, that is the Km, that is Michaelis constant. Km, it is this value, wherein the velocity has reached half its value. Now moving on to the classification and no. Yes?
Ma'am, can they ask, like, what kind of questions will they ask us, like, related to these graphs?
Related to the graphs. See, here they can ask you, for this, they can ask you where is the activation site decreasing, where is it increasing, and for example, they did not label anything. They can ask you what is on the y-axis, what is on the x-axis. Apart from that, they can also ask you out of these two, which one is enzyme mediated and which one is not. Okay. Now talking about these, they can ask you, these are the effects how pH and temperature affect it. Okay, so again, the denaturing and everything can be mentioned here. Okay, and as far as this goes, nothing can be asked of this, but you should know that when the maximum velocity reaches half its value, that point is known as Michaelis constant. Clear?
Yes, ma'am.
All right, now talking about the classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the type of reactions they catalyze. Enzymes are divided into 6 classes, each with 4 to 13 subclasses, and named accordingly by a 4-digit number. So what are the classes of enzyme? There is oxidoreductase or dehydrogenases, transferases, hydrolases, lyases, isomerases, and ligases. You must remember the class of the enzyme. Now, enzymes which catalyze, now from here, the question that can be asked, like, what is the name of the enzyme which catalyze oxidoreduction between two substrates, S and S dash. So you should know that it is oxidoreductases kind of enzymes or dehydrogenases. Transferases, what they do is that these catalyze a transfer of a group G other than the hydrogen between a pair of substrates. So it is responsible for the transfer of a group. Hydrolases, what it does the breaking part, like hydrolysis of ester, hydrolysis of ether, peptide, glycosidic bonds, then carbon-carbon single bond, carbon halide bond, and phosphate nitrogen bonds. So these are responsible for the lysis, that is the breakdown. Now, lyases, they are responsible for the removal of groups from substrates by mechanism other than hydrolysis. Okay, so, and apart from that, it leaves the double bond. So, for example, carbon-carbon single bond were there and it had 2 functional groups. Those functional groups will be separated, plus the carbon-carbon will develop a double bond. That is the function of lyase. Now, isomerases. These catalyze interconversion of optical, geometrical, or positional isomers. Lastly, there is ligases. We know that they are responsible for joining. They are glue-like enzymes. So they are responsible for the linking together of two compounds. For example, enzymes which catalyze joining of carbon and oxygen, carbon and sulfur, carbon and nitrogen, phosphorus and oxygen bonds. So this carbon and with all the other elements, this bonding, the joining of the two bond is because of the ligase enzyme. Now we'll talk about the cofactors. Now enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which non-protein constituents called cofactors are bound to the enzyme to make the enzyme catalytically active. Meaning these are the substances, cofactors are the ones that enhance the enzyme activity. In these instances, the protein portion of the enzyme is known as the apoenzyme. Three kinds of cofactors may be identified. One is the prosthetic group, second is the coenzyme, and third is the metal ion. So cofactors are of three types. Prosthetic groups are organic compounds. Remember, this is important. They are organic, naturally occurring, and are distinguished from other cofactors in that they are tightly bound to the apoenzyme. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide into water and oxygen, heme is the prosthetic group. Meaning here, the enzyme will, the peroxidase or the catalase enzyme would act better, will show better catalytic activity because of the prosthetic group, that is the heme. Now talking about coenzymes, the second type of cofactor. These are also organic compounds, but their association with the apoenzyme is transient for a short period of time. Here it is tightly bounded for a long period of time, usually occurring during the course of catalysis. Furthermore, coenzymes serve as cofactors in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins, such as coenzyme nicotinamide adenine dinucleotide (NAD) and NADP dinucleotide phosphate contain the vitamin niacin. And what are they? These are the coenzymes. Now, a number of enzymes require metal ions for their activity, which form coordination bonds with side chains at the active site, and at the same time form one or more coordination bonds with the substrate. So zinc is a cofactor for the proteolytic enzyme carboxypeptidase. So again, a metal ion can also act as a cofactor, thereby increasing or enhancing the catalytic activity. Now, catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in catalytic activity of the enzyme. So this much is clear, students?
Yes, ma'am.
Any doubts up until now?
No, ma'am.
Okay, so with this, oh, good. So with this, we have actually completed the chapter that is biomolecules. And with this, we have actually completed the class 11th portion entirely. So now you people do one thing, take 5 minutes, read the chapter, like read the topics and everything and see if you have any doubts anywhere. And after this, we'll start molecular basis of inheritance, okay? You take your time, read it, then we'll start a new chapter. Yes, students, any doubts?
No, ma'am.
Okay, so now we're going to start with the molecular basis of inheritance. Just one second. These look like my notes. Just one second. Is my screen visible?
Yes, ma'am.
Yes, ma'am.
All right, so we're going to talk about the molecular basis of inheritance. Now, remember that this whole entire chapter consists of how DNA works, how there are certain bonds present, what are the process of gene expression, and everything that goes along with this. Now, when we were talking about the this chapter, biomolecules, we talked about the nucleic acids, right? Now, the nucleic acids in our body are of two types. There is deoxyribonucleic acid, and then there is ribonucleic acid, DNA and RNA. Now, DNA is the genetic material for maximum number of organisms, right? For maximum number. Now, DNA acts as the genetic material in most of the organisms. RNA, though it also acts as a genetic material in some viruses, but it mostly functions like a messenger. RNA has two additional roles as well. It functions as an adapter, structural, and in some cases as a catalytic molecule. This is the reason why it is not the permanent, you can say, genetic material, because it has catalytic roles. It has functions that the genetic material must not have. It is not that stable as compared to DNA. Now, okay, we know how nucleotides are made up, right? They are made when the nucleoside combines with the phosphate group, right? Now, and the way these monomer units are linked to form the nucleic acid polymers. Now, in this chapter, we are going to discuss the structure of DNA, its replication, the process of making RNA from DNA, transcription, the genetic code that determines the sequences of amino acid in proteins, the process of protein synthesis, that is translation, and the elementary basis of their regulation. Now, the determination of complete nucleotide sequence of human genome during the last decade has set in a new era of genomics. In the last section, the essentials of human genome sequencing, all of these things will be discussed. Now, let's start with the DNA. We know that it is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as the number of nucleotides or a pair of nucleotides referred to as base pairs that are present in it. So the DNA, the length of the DNA can be actually calculated or can be defined with the help of the base pairs. Now, this is also the characteristic of an organism. The length of the DNA depends from organism to organism. For example, a bacteriophage known as, what is this symbol known as? Delta 174 has 5,386 nucleotides. Bacteriophage lambda has 48,502 base pairs in it. E. coli has 4.6 into 10 to the power 6 base pair, and haploid content of human DNA has 3.3 into 10 to the power 9 base pair. This is again, it is a characteristic feature. Now, this human DNA, this length or the content for haploid human DNA, it is for all of them, not just one, for every single, you can say, human. It does not have any exception to it. Now talking about the structure of polynucleotide chain. Let us recapitulate the chemical structure of a polynucleotide chain, DNA or RNA. A nucleotide has three components. The nitrogenous base, that can be a purine or a pyrimidine. A pentose sugar. Pentose sugar means 5 carbon sugar, and it is ribose in the case of RNA and deoxyribose for DNA. And then there is a phosphate group. And when all of them are present together, we'll call it as the nucleotide. Now, there are two types of nitrogenous bases, that is purine, which is adenine and guanine, and then there is pyrimidine, that is cytosine, uracil, and thymine. Now, cytosine is common for both DNA and RNA, and thymine is present only in DNA. Uracil is present only in RNA at the place of thymine. Now, a nitrogenous base is linked to the OH, hydroxyl of the first carbon of the pentose sugar through an N-glycosidic linkage. So, the linkage between a nitrogenous base to the sugar is a glycosidic linkage, and it forms a nucleoside such as adenosine, adenosine, deoxyadenosine, guanosine or deoxyguanosine, depending upon the sugar that is there. Now, apart from that, when a phosphate group comes linked to the nucleoside, at what position? So, at the hydroxyl of fifth carbon of a nucleoside. Now, which bond? Phosphoester linkage. A corresponding nucleotide is formed. Two nucleotides are together linked like 3' and 5' phosphodiester linkage to form a dinucleotide. So the name of the bonds are very important. They can be mentioned in the exam in the questions as to which bond is present between the nucleoside, which one is present in the nucleotide. All of these things will be very crucial as far as the, you can say, assertion reason kind of question goes, and the statement wise question goes. This is very important. Now, more nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has at one end a free phosphate moiety, meaning at the 5' end of the sugar, there will be a phosphate group which will be free, and which is referred to as 5' end of the polynucleotide chain. 5' will contain the phosphate group attached onto it. Now, similarly, at the other end of the polymer, the sugar has 3 OH, like it has at the third prime carbon at this side, like just the opposite, it will have a 3 hydroxyl group, which is referred to as the 3' end of the polynucleotide chain. So, one polynucleotide chain, it has a 5' end and it has a 3' end. Now, the 5' end has the phosphate group and the 3' end has the hydroxyl group. Again, a very important line. The backbone of a polynucleotide chain is formed due to sugar and phosphates. The nitrogenous base linked to the sugar moiety projects from the backbone. Now, in RNA, every nucleotide residue has an additional OH group present at the 2' position in the ribose. Also, in RNA, the uracil is found at the place of the thymine, that is 5-methyl uracil, another chemical name for thymine. Now, DNA as an acidic substance. Now, we are talking about how the DNA was termed as an acidic substance. Why is it not basic? So, it was actually identified by Frederick Masker. He was the person who told that, okay, DNA, it is an acidic substance. He named it as nuclein at that point of time. It was not named as DNA. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a long period of time. Now, we know that DNA is a really long molecule, right? Because of that very reason, what happens is that it is very hard to find out how the DNA structure was at that point of time. So what they did, the structure was unknown for a really long period of time. Now, it was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by the Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. So who gave the double helical structure of DNA? It was Watson and Crick, very famous for it, and this is very important to know also. Now, one of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. However, this proposition was also based on the observation of Erwin Chargaff that for a double-stranded, the ratios between adenine and thymine and guanine and cytosine are constant and equals 1. So this is again Erwin Chargaff's rule. It becomes like a rule that the ratio will equal to 1. Now, the base pairing confers a very unique property to the polynucleotide chain. They are said to be complementary to each other, and therefore, if the sequence of bases in one strand is known, then the sequence in other strand can also be predicted, because we know that adenine will always pair up with what? Tell me. Adenine pairs up with
Thymine.
thymine. And how many bonds are there in between?
2.
Cytosine pairs up with
Guanine.
guanine, and 3 bonds are there. Same goes for here, same goes for here. So this is a fixed thing. Now, if we know this side, we can know this side. So that is why this is the complementarity that helps us to know the sequence of the DNA. Also, if each strand from a DNA, let us call it as a parental DNA, it acts as a template for the synthesis of a new strand. The two double-stranded DNA, let us call them as daughter DNA, thus produced, would be identical to the parental DNA molecule, because we know that the DNA is a double-stranded molecule. Now, when this will open, each of them will act like a parental DNA or a template DNA, according to which new DNA strand will be formed. Because of this, the genetic implications of the structure of DNA became very clear. The salient features of double helix structure of DNA are as follows. It is made up of two polynucleotide chains where the backbone is constituted by sugar phosphate and the bases project inside. Like all of these salient features are very important to remember. Okay. So it is made up of two polynucleotide chains where the backbone is constituted by the sugar phosphate and the bases project inside. The two chains have anti-parallel polarity. It means that if one chain has the polarity from 5' to 3', the other will have the polarity from 3' to 5'. And this is a said thing, it cannot change ever. Okay, this is what happens. If the polarity at one strand is 5 to 3, the other will be 3 to 5. It has to be like that. Now, the third is that the bases in two strands are paired through hydrogen bonds, forming the base pairs. Adenine forms two hydrogen bonds with thymine from opposite strand and vice versa. Like the thymine will also form two bonds with adenine. Similarly, guanine is bonded with cytosine with three hydrogen bonds. As a result, always a purine comes opposite to the pyrimidine. One purine will bind with one pyrimidine, always. This generates approximately uniform distance between the two strands of the helix. And remember that these are the hydrogen bonds. Now the two chains are coiled in a right-handed fashion, meaning the helix is like this. It's not left-handed. Okay. Now the pitch of the helix is 3.4 nanometer, the pitch of the DNA helix. And there are roughly 10 base pairs in each turn. Consequently, the distance between a base pair in a helix is approximately 0.34 nanometer. The plane of one base pair stacks over the other in a double helix. This, in addition to hydrogen bond, confers stability to the helical structure. So, two things are actually providing stability to this helical structure. One is that the base pairs are stacked upon each other, and second, the presence of hydrogen. Now, the proposition of a double helix structure for DNA and its simplicity in explaining the genetic implication became revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology, which states that the genetic information flows from DNA to RNA to protein. This is the flow of genetic information, and that is how it happens. And this central dogma in molecular biology, it was proposed by Francis Crick. Now, the DNA will form more DNA by DNA replication. Now, the DNA will form mRNA by the process of transcription. The mRNA would form protein by the process of translation. So, this is how the genetic information flows, and this is the pathway that it will follow. Now, moving forward, in some viruses, the flow of information is reversed. Now, what happens is that from the RNA, DNA can be made by the enzyme that is known as reverse transcriptase. Okay, and this is known as reverse central dogma, and this is for the viruses. Moving forward, talking about the packaging of DNA helix. Up until now, you people have understood everything?
Yes, ma'am. Yes, ma'am.
Any doubts?
No, ma'am.
All right, now we'll talk about the packaging of DNA helix. Taken the distance between two consecutive base pairs as 0.34 nanometer. If the length of the DNA double helix is in a typical mammalian cell is calculated simply by multiplying the total number of base pairs with distance between the two consecutive base pairs, it comes out to be approximately 2.2 meters. So our DNA, one DNA is 2.2 meters long, a length that is far greater than the dimension of a typical nucleus, and the nucleus is in the size of 10 to the power minus 6 meter. Imagine how tiny a nucleus is and how long a DNA is. So and we know that in the eukaryotic organisms, the DNA is present in the nucleus only. It is not present elsewhere. And how does that happen? Why does that happen? It's because now how can the DNA fit into the nucleus? That comes the question. Like if the DNA is 2.2 meters long and the nucleus is just 10 to the power minus 6 meter. So it means that the DNA is folded to fit inside, right? Now in prokaryotes such as the E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. DNA, being negatively charged, is held with some proteins that have positive charge in a region which is termed as nucleoid. That okay, we understand that okay, prokaryotic organisms, they do not have a nuclear membrane surrounding the nucleus. Their genetic material is naked. But even after that, the DNA is not present throughout the cell. It is concentrated in an area wherein we know that the DNA is negatively charged. So that negatively charged DNA is held together with positively charged proteins in a region which is nucleoid. Now the DNA in nucleoid is organized in large loops held by proteins. Now for eukaryotes. This organization is much more complex. There is a set of positively charged basic proteins known as histones. A protein acquires charge depending upon the abundance of amino acid. So if in a protein there are more, we know that protein is a heteropolymer. So more basic amino acid if there will be, then the protein will be positive. Now histones are rich in the basic amino acid residues, that is lysine and arginine. Both the amino acid residues carry positive charges in their side chain. Histones are organized to form a unit of 8 molecules which are known as histone octamer. 8. Now the negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called as the nucleosome. So when the DNA wraps around the histone, it makes a nucleosome. Now a typical nucleosome contains 200 base pairs of DNA helix. Nucleosome constitute like the repeating unit of a structure in nucleus known as the chromatin. Thread-like stained or the colored body seen in the nucleus. These are chromatins. Now, the nucleosomes and chromatins, they are seen as beads on string structure when viewed under the electron microscope. What is seen? The chromatin. Okay, the chromatins and the nucleosomes. So they look sort of like a bead that has been attached onto a string or a string with several beads. Okay, now the beads on string structure in chromatin is packed to form chromatin fibers that are further coiled and condensed as metaphase stage of cell division to form chromosome. So starting off, we have DNA plus histones making the, first of all, histones making the histone octamer. Then DNA wrapping around the positively charged histone making nucleosome. Nucleosome being present together with chromatin. And then lastly, from the chromatin, chromosomes are formed. Now, the packaging of chromatin at high level requires additional set of proteins that are collectively referred to as non-histone chromosomal proteins, NHC. Now, in a typical nucleus, some region of chromatin are loosely packed and they are lightly staining. So they are referred to as euchromatin. Now, euchromatin, they are transcriptionally active. They are loosely packed and that is why they lightly stain. Now, the chromatin that is more densely packed and stains dark are known as heterochromatin. They are transcriptionally inactive. This part is very important. Now, this is the introductory part as to how, what, why, and everything goes along. Now, talking about how the genetic material was found, how people finally came to the conclusion, and how now we know that DNA is the genetic material. So, there is a long process that goes behind it. Up until now, do you have any doubt?
No, ma'am.
No, ma'am.
All right, now talking about the search for genetic material. Even though the discovery of nuclein by Friedrich Miescher and the proposition for principle of inheritance by Mendel were almost at the same time. But the DNA acts as a genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Mendel, Sutton, and T.H. Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most cells. Now we know that because of these scientists, it was found out that the chromosomes are situated in the nucleus of the cells. Again, what do the chromosomes carry was unknown. Now talking about the transforming principle. In 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumoniae, the bacteria that causes pneumonia, witnessed a miraculous transformation in bacteria. During the course of his experiment, a living organism, that is bacteria, had changed in the physical form. When Streptococcus pneumoniae, that is pneumonococcus bacteria, was grown on a culture plate, it produced smooth, shiny colonies, while others produce rough colonies. This is because the S strain bacteria have a mucus polysaccharide coat, while the R strain do not. Now, the S strain were the ones that actually caused the disease. That is why they produce the smooth and shiny colonies. The R strain did not cause any disease. Now, mice, at that point of time, mice was used for any kind of, you can say, human-related discovery or what do you call, experiment. Now, mice infected with the S strain or the virulent means disease causing, potentially causing it to die from pneumonia infection, but the mice infected with the R strain do not develop pneumonia. So when the R strain was injected into the mice died. When the R strain was injected, then the mice lived. Now, Griffith was able to kill bacteria by heating them. He observed that the heat killed S strain bacteria injected into the mice did not kill them. So when the S strain, that is the virulent strain that actually causes pneumonia, when that bacteria was heat killed and then inserted into the mice, it did not cause any kind of disease or the mice did not die. Then the heat killed S strain along with the living R strain was inserted into the mice died. Now this was very shocking to them. So what happened? He concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Some transforming principle transformed them from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide core and become virulent. Now, because something from the heat killed S strain made the R strain become virulent. So this must be due to the transfer of genetic material. However, again, what was the genetic material? Still not known. Now, the biochemical characterization of transforming principle. Now, prior to the work of Oswald Avery, Colin McLeod, and Macklin McCarthy from 1933 to 44, the genetic material was thought to be a protein. People thought that, okay, whatever is the genetic material, we don't know the chemical nature, but we think that it is protein. They worked to determine the biochemical nature of transforming principle in Griffith's experiment. So now Griffith had done his experiment. Now a question can be asked like who did the biochemical characterization? So you should know Avery, McLeod and McCarthy did it. Now what they did is that they purified biochemicals such as protein, DNA, RNA from the heat-killed S cells or the S which is the smooth cells to see which ones could transform the living R cells into the S cells. They discovered that DNA alone from the S strain bacteria caused the R strain to become transformed. So they did test on each of the biochemicals that could be there, proteins, DNA and RNA. So they did a test on all of them. Now they discovered protein digesting enzymes that are known as proteases and RNA digesting enzymes that are RNases. Now they did not affect the transformation. They saw that when these proteases are the one that will destroy or digest the protein. Same goes for RNAs. So they said that, okay, whilst doing the transformation experiment, let us put these enzymes. Now, when these enzymes were put, nothing happened to the transformation. But when DNAs was put, then the transformation did not happen. So this suggested that the DNA caused the transformation. They concluded that DNA is the hereditary material, but again, not all biologists were convinced at that point of time. Any doubts up until now? Hmm?
No, ma'am.
No, ma'am.
Okay. All right. Now, talking about the genetic material is DNA. The unequivocal proof that DNA is the genetic material came from the experiments of Alfred Hershey and Martha Chase. Hershey and Chase experiment in the year 1952. They worked with viruses that infect bacteria that are known as bacteriophages. The bacteriophage attaches to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particles. Hershey and Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria. They grew some viruses on a medium that contained radioactive phosphorus and some others on the medium that contained radioactive sulfur. Now, viruses grown in the presence of radioactive phosphorus contain radioactive DNA, but not radioactive protein because DNA contains phosphorus, but protein does not. Protein has sulfur in it. DNA has phosphorus. So the DNA that was grown in the radioactive phosphorus, they took up the radioactive phosphorus, but it did not take the sulfur. Like vice versa. Now, similarly, virus grown on the radioactive sulfur contain the radioactive protein, but not radioactive DNA. because DNA does not contain sulfur. Radioactive phages were allowed to attach to E. coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. The virus particle was separated from the bacteria by spinning them in a centrifuge. So, centrifugation happened. The bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that the DNA was the material that was passed from the virus to the bacteria. But the bacteria that were infected with the viruses that had radioactive protein were not radioactive in nature. This indicated that the protein did not enter the bacteria from the viruses. It was the DNA. So with this experiment, they had concluded that DNA is the genetic material. Now, properties of genetic material, that is DNA versus RNA. Why DNA is only the genetic material? Why not RNA? This is what we will talk about right now. From the foregoing discussion, it is clear that the debate between protein versus DNA as the genetic material was unequivocally resolved from the Hershey-Chase experiment. DNA won there. It became an established fact that it is DNA that acts as a genetic material. However, it became clear that in some viruses, RNA was the genetic material. For example, the tobacco mosaic virus, the QB bacteriophage, the AIDS virus, that is HIV, that also has RNA as the genetic material. So, answer to some of the questions such as why DNA is the predominant genetic material, whereas RNA performs dynamic functions of a messenger, adapter, then even after this, why only DNA is the genetic material? Now, what are the qualities that the genetic material must have? It should be able to generate its replica, meaning it should be able of replication. It should be stable chemically and structurally. So it should be structurally and chemically stable. It should provide the scope for slow changes, very slow, not instant and quick. So mutation scope should be there. And it should be able to express itself in the form of Mendelian characters. Now, if one examines each requirement one by one, because of rule of base pairing and complementarity, both the nucleic acids have the ability to direct their duplication, meaning they can replicate. The other molecules cannot, proteins cannot. So the genetic material should be stable enough not to change with different stages of life cycle, age, or changes in the physiology of organisms. Stability as one of the properties of genetic material was very evident in Griffith's transforming principle. It said that heat which killed the bacteria at least did not destroy some of the properties of genetic material. This now can easily be explained in the light of DNA, that the two strands being complementary, if separated by heating, come together when appropriate conditions are provided. Further, the 2' OH group, the 2' hydroxyl group that is there, at every nucleotide in RNA, it is a reactive group and it makes the RNA labile and easily degradable. Again, a very important line this is. The 2' hydroxyl group that is present in every nucleotide in RNA, this is a reactive group. Because of the presence of this group, the RNA becomes labile and easily degradable. It is very reactive. Okay, and RNA is also now known to be catalytic in nature. Hence, it is reactive. So, because of all of these things, it is not stable. It is degradable. So, it cannot be the genetic material. Now, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acids, the DNA is a better genetic material. In fact, the presence of thymine in the place of uracil also adds the stability to the DNA, more and more stability. Both DNA and RNA are able to mutate. In fact, RNA being unstable mutates at a faster rate, which is not something that we require as a genetic material. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. RNA, that is why viruses can change so quickly because they have a scope for mutation much faster as compared to DNA. RNA can directly code for the synthesis of proteins, hence can easily express the character. DNA, however, is dependent on RNA for the synthesis of proteins. The protein synthesizing machinery has evolved around the RNA. So, we know that both RNA and DNA can function as genetic material, but DNA being more stable is preferred for the storage of genetic information. For the transmission of genetic information, RNA is better. For storage, DNA is better. It is stable. But if you want to transmit it, if you want to express the information, then RNA would be better. Now talking about RNA world. From foregoing discussion, an immediate question becomes evident, which is the first genetic material. It shall be discussed in detail, but we should know that RNA was the first genetic material. There is now enough evidence to suggest that essential life processes such as metabolism, translation, that is process of formation of protein, and splicing evolve around RNA. RNA used to act as a genetic material as well as a catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalysts and not by protein enzymes. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA evolved from RNA with chemical modification that makes it more stable. DNA being double-stranded and having complementary strands further resists changes by evolving a process of repair. So again, even though RNA was the first genetic material, but then DNA was formed off of RNA and it was much more stable. Any doubts up until now?
No, ma'am.
No, ma'am.
Everything is clear till here?
Yes, ma'am.
All right. Okay. Okay, now we'll talk about the replication. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism. They told us about the replication. This is the statement that they passed out. And they said that the scheme suggested that the two strands would separate and each of those two strands would act as a template for the synthesis of new complementary strands against each of them. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication. Why is it semi-conservative? Because out of the two DNA, out of the two strands in a DNA, one is the parental DNA and the other one, the newly synthesized one. So that is why it is termed as semi-conservative. Now again, the experimental proof to tell us, is it really semi-conservative? Yes or no? Now it is proven that DNA replicates semi-conservatively. It was shown first in E. coli and subsequently in higher organisms such as plants and human cells. Matthew Messelson and Franklin Stahl performed the experiment to tell us whether the DNA is semi-conservative in nature or not. DNA replication actually. They grew E. coli in a medium containing the you can say heavier isotope of nitrogen. So they took NH4Cl, but the N here was the heavy isotope. Now, the result was that the N15 was incorporated into the newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in cesium chloride, CaCl. Density gradient. Remember that this N15 is not a radioactive isotope and it can be separated from the N14 only based on density. So, because this is heavier, right? It is not radioactive, but it is heavier in molecular weight. So, we can distinguish whether the nitrogen is N15 or N14 depending upon the density. Now, then they transferred the cells into a medium with the normal nitrogen, that is NH4Cl, that is N14. And they took samples at various definite time intervals as the cells multiplied and extracted the DNA that remain as double-stranded helices. The various samples were separated independently on cesium chloride gradients to measure the densities of DNA. So what was found? Initially, the one that had the N15, the heavier isotope, it was heavy. The one that had the N14, it was the light. But then the one that had both of them, N14 and N15, that was a hybrid, their weight was in the middle. Thus, the DNA extracted from culture one generation after the transformation from N15 to N14 medium, that is after 20 minutes, E. coli divides in every 20 minutes, it has a hybrid or intermediate density, the density in the middle. The DNA extracted from the culture was another generation, that is 40 minutes or the second generation. Now was composed of equal amounts of this hybrid DNA, that was the light. Now if E. coli was, now see, they took 20-minute interval and after each interval they saw the weight of the DNA. And with that being said, it was because of this hybrid, meaning in the middle, after the centrifugation, it was found that the light and the heavy to be existing together. That tells us about the semi-conservative nature. Now, very similar experiment involving the use of radioactive thymidine to detect the distribution of newly synthesized DNA in the chromosomes was performed on Vicia faba, that is faba beans, by Taylor and his colleagues in 1958. This experiment proved that the DNA in chromosomes also replicate semi-conservatively. So up until now, we have understood and we have realized that the DNA replication is semi-conservative in nature. And girls, what is the meaning of semi-conservative? What is the meaning of semi-conservative?
One strand will be like the parental and the other one will be female.
Yes. Okay. Just give me 1 second. Students, I'm just coming in 2 minutes. Okay, so the DNA being semi-conservative is clear to you people?
Yes.
Yes, ma'am.
Okay, good. Are you finding the chapter easier now that I'm teaching, or is it still difficult?
It's easier a bit.
It's easier? Now you're understanding?
Yes, ma'am.
Okay, that makes me really happy. All right, now the machinery and the enzymes. Now we'll talk about how the DNA replication happens. What are the things that go behind? Now you know the basic concept. If the DNA is stranded and it is helical, so these helix, for example, if this is the helix, during the time of replication, it will open like this, and each of them will synthesize their own DNA, and this will be the parental DNA. Okay. Okay. In living cells, such as E. coli, the process of replication requires a set of catalysts, that is enzyme. Now, again, as I told you, that in biology, any reaction, no matter what it will require enzymes. Now, the main enzyme is referred to as the DNA-dependent DNA polymerase. This is the enzyme that is used for the replication process. Now, what does it do? It uses a DNA template to catalyze the polymerization of the deoxynucleotides. Polymerization, you people understand, it means creating multiple copies. Now, these enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. E. coli that has only 4.6 into 10 to the power 6 base pair, compare it with human whose diploid content is 6.6 into 10 to the power 9 base pair. And both of them complete the process of replication within 18 minutes. Imagine having 6.6 into 10 to the power 9. Are you getting it? 6.6 meaning 10, 9 times zeros. That many base pairs are actually polymerized in just 18 minutes. This means that the average rate of polymerization has to be approximately 2000 base pair per second. It is polymerizing 2000 base pairs in 1 second and each second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy. Any mistake during replication would result into mutation. Furthermore, energetically, replication is very expensive process. It requires a lot of energy. Now, deoxyribonucleoside triphosphate serve as dual purpose. In addition to acting as substrates, they provide energy for polymerization reaction. The two terminal phosphates in deoxynucleoside triphosphates are high energy phosphate, same as in the case of ATP. Like how adenosine triphosphate is there, same way for replication, because it requires a huge amount of energy, like higher than the normal. So ATP will not suffice. So a greater molecule will be required, and that particular one is deoxynucleoside triphosphate. They have high amount of energy that serves for the replication process. In addition to DNA-dependent DNA polymerases, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length, because we know that the DNA is 2.2 meters, right? So 2.2 meters long DNA, if we try to, you can say, open it all at once, it will be so impossible. Like, for example, people that have long hair. Now, if they have made plates, when they want to open their hair, they do it slowly, slowly, right? They can't just do it like this from the base and then it will be open. That's not possible because the length is long. Same thing happens for DNA. So DNA does not open the entire length all at once. It opens part by part, part by part. So since the two strands of DNA cannot be separated in its entire length due to very high energy requirement, the replication occurs within a small opening of DNA helix that is known as the replication fork. For example, we know that DNA is 2.2 meters long. Again, maybe it will open at this point of time. So this area will be called as the replication fork, meaning at that area only new DNA templates or new DNA is synthesized. Now the DNA-dependent DNA polymerases catalyze polymerization only in one direction, that is from 5' to 3'. This creates some additional complications at the replicating fork. Consequently, on one strand, the template with polarity that is 3' to 5', the replication is continuous, while on the other hand, from 5' to 3', it is discontinuous. Continuous replication means, again, the parental strand, a continuous piece of DNA is synthesized. Discontinuous means small pieces are synthesized. Now, the discontinuously synthesized fragments, now it is in the type of fragments. Now, they are joined. Now, obviously, for a new DNA to be formed completely, it has to be formed and it has to be formed entirely. So, that is done with the help of the DNA ligase. Now, we have studied that ligase is that class of enzyme that joins bonds. So, again, for this, DNA ligase is important. Now, DNA polymerases on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. There is a definite region in E. coli DNA where the replication originates. Such region is known as origin of replication. Just like I just drew here to make you understand that, okay, this part is opening, but that is not how it happens practically. It cannot open anywhere and start synthesizing. It will open at a certain place that is known as the origin of replication. Now, it is because of the requirement of the origin of replication that a piece of DNA, if needed to be propagated during recombinant DNA procedures, it requires a vector. The vectors provide the origin of replication. Now, without the origin of replication, the replication will not take place whatsoever. Further, not every detail is understood well of like for replication. In eukaryotes, the replication of DNA takes place at the S phase of the cell cycle. S phase comes in which phase of the cell cycle? Quick. Which phase of the cell cycle?
Interphase.
Interphase. Very good. The replication of DNA and cell division cycles should be highly coordinated. A failure in cell division after replication results into polyploidy. Polyploidy is one cell with multiple nuclei that fail to form the cell wall. The cell did not divide and one cell has more than one nuclei. So, with that being said, not actually more than one nuclei, but multiple DNA strands are present, and that is a polyploidy situation, which is a chromosomal anomaly. Chromosomal anomaly means chromosomal abnormality. So this is how the replication process happens. And see, this is a replication fork. And here, see, as from 5' to 3', there is the discontinuous strands. And then here is the continuous strand. These strands, they are also known as the Okazaki fragments, and they will be joined by the DNA ligase. So DNA replication is clear, students?
Yes, ma'am.
So what are all the things that are needed for DNA replication? See?
DNA polymerase.
DNA dependent DNA polymerase. There are many DNA polymerase that are RNA dependent as well. So remember that to mention that it is DNA dependent DNA polymerase. Very good. Then after that,
Replication fork.
okay, but initially what do we need? We need origin of replication, without of which the replication will not happen. Now we need a replication fork. Yes, very good. Then we need DNA dependent DNA polymerase. Now we also need the DNA ligase. So all of these things are very important for DNA replication process. Now moving forward, let us talk about transcription. Now the formation of RNA, more importantly messenger RNA, mRNA from DNA is known as transcription. The process of copying genetic information from one strand of DNA into RNA is termed as transcription. Here also the principle of complementarity governs the process of transcription, except the adenosine complements now forms base pair with uracil instead of thymine. Because normally adenine forms a bond with thymine, but if we are talking about, this is for DNA. Now, if you are talking about RNA, then this adenine will form bond with uracil because in RNA, the thymine gets substituted by uracil for RNA. However, unlike in the process of replication, which once set in, the total DNA of the organism gets duplicated. In transcription, only a segment of DNA and only one of the strands is copied into the RNA. This necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. So transcription, you people have understood that it is how the DNA copies the information to make the RNA, more importantly, the mRNA. During this process, the adenine forms base pairs with uracil, and apart from that, once the transcription starts, it is only done for one segment, not the entire length of DNA. Okay, and as far as this thing goes, the DNA replication, once it starts, the whole entire DNA gets replicated. Any doubts up until now?
No, ma'am.
Sure?
No, ma'am.
Yes, ma'am.
Okay, okay, very good. All right, then let's just keep it till here only for today. Are you people tired? Because technically we are doing 2 chapters, isn't it? Biomolecules is done. Now we are almost in the middle of molecular basis. So you tell me, are you people tired?
A bit.
A bit? Okay. Okay, we'll keep it till here only for today. Now in the next class, we'll complete transcription. We'll also discuss about translation, different types of RNA. Now this different types of RNA is very important. And there are so many things that we yet have to discuss. So up until now, do you have any doubts?
No, ma'am.
Sure?
Yes, ma'am.
Oh, all right. Then let's just keep it till here only. Now in the next class, we'll continue from transcription. Okay? Until then, read biomolecules, because biomolecules is again a really tough chapter in terms of its language. Many a times it's very difficult to understand what the question is saying. So pay attention to that. Practice the question. That's the most important thing. Now you're like only how many days away from NEET? It's like so close, right? So practice as many questions as you can. Read as much NCERT as